Okay, so I think we should start the live session. Let me just share my screen. Uh, and someone, please let me know whether the screen and the slide share is visible and whether I am clearly audible or not. Okay, I see in the chat, visible and audible. Okay, fine. Okay, so again, welcome all of you to the course Bioinformatics Algorithms and Applications. This NPTEL course is instructed by Professor Michael Gromiha. And today, for the week 2 live tutorial session, I will take it. So, my name is Sunanda Samanta, a PhD scholar at IIT Indore, and I work at the Computational Biophysics group here at the Department of Biosciences and Biomedical Engineering at IIT Indore. So, uh, so the last week we went over some basic concepts of bioinformatics, right? What are the broad application areas, uh, and fundamentals of like sequence-based parameters and so on, right? Essentially, like covering an overall uh, aspect or overall idea about bioinformatics, right? And for this week's course lecture, I think there was some protein function introduction, right? And the secondary structures and some basic biochemistry of amino acids, uh, and then some sequencing concepts like how to do sequence alignment and dot plots and those kinds of things, right? So we'll briefly cover some of those concepts. For the last class when I took the tutorial, so the last Friday, I did not cover some of the earlier concepts as well, like databases and database management systems and so on. So some of those I would also like to cover in this class. And again, so the tutorial sessions that will be taken by the TAs, so all three of those will cover broadly the same area. So there will be other two tutorial sessions done by two other different PhD scholars. So you can also join them if you have the time available, right? Okay. Uh, okay, fine. So let us start today's tutorial session. The first thing I would like to cover is some basic concepts about databases, uh, because that is one of the big parts, in the broad field of bioinformatics. So can somebody quickly remind me why do we need databases? What function do they serve to the overall scientific community, or any sort of field of research uh, where databases might be important? Any idea? So you can unmute or you can type in the chat as well. So databases, as you know, they are basically an organized collection of data, right? Now imagine in some field of science, right? Let's say research is going on all over the world by different research groups, like across the globe. And throughout their research, they are collecting some data, right? They're analyzing those data and they're publishing the data. That's fine. And a lot of important data points, if you think about it, those are available uh, throughout the landscape of the scientific field, right? Now, what happens is that whenever you, for example, start a new research study, like within a particular field, so you have to go through the literature, you have to collect all the previously available information, all the data that is available through the previous experiments done by the other research groups and so on. So that takes a lot of time, and some data points they are so relevant in the broad fields, is that there will be a lot more convenience and time reducing uh, thing if those data points or the data from the previous research experiments, those are collected and organized in a single database or server, right? So what will happen is that this will allow anybody who is interested in that field, or who is any research group who wants to study something on that field. So they will be quickly able to get their hands on the data, the relevant data, through this organized databases, right? This organized structure. And they will be able to extract the relevant information, what is needed by them, very quickly and efficiently. And this will essentially reduce the time of doing this literature survey and all by a lot, and reduce a lot of headaches essentially, right? In the initial phase of the research. So that's very important. So that's why databases are, as Professor Gomi also mentioned in these lectures, that creating databases and collecting data and everything, so that is uh, essentially very important in the broad field of bioinformatics as well, where we collect the biological data, the data that is relevant to different biological experiments uh, in the form of databases, right? So, any examples? Can somebody give an example of any biological databases? I think we went over some of these databases. They could be related to genetic information, or protein structural information, or protein sequence information, anything like that. Yeah, DNA sequences. But can you name some of the databases that perhaps contain DNA sequence?
UniProt,
Yeah, UniProt is basically protein sequence. So I think we have heard of
DBJ.
yeah, DDBJ, EMBL, GenBank, all of this. So we'll just go quickly through some of those. Okay. So basically, to sum up what I said is that a database is basically an electronic repository which is designed to store or organize data. Not only that, but it has to facilitate easy retrieval based on the various search criteria. So those are some of the features that are included in database uh, for it to be effective, essentially. And it comprises both the hardware and software components. And the primary goal is to not only store the data in a structured format, but also have different fields such as names, phone numbers, addresses. This is a like generic database you can think of, which can enable quick retrieval of some information based on the criteria that you give as a query, right? Second, you can query the database by specifying particular values, uh, and then some of the key characteristic features will include, first of all, the content, the base data that is stored in the database, then ontology, the list of valid terms, their definitions, then logical structure, so that is the interrelationships among data, which is also known as schema, the data format, the retrieval methods, analysis programs, and also links to other external information resources such as other databases, original publications, tutorials, etc. So basically, it has to be very well annotated and highly integrated with the outside resources as well, right? Outside of the database. So that's some of the crucial features of a database. Now, if you talk about the biological databases and the categories that are covered by the biological databases, so based on their types of contents, they are primarily divided, or they're basically divided into three types of databases. Firstly, you can think of this primary database, another one is secondary database, and some specialized databases. Okay. So primary databases, they basically store original biological data such as raw sequence or raw structured data submitted by the scientific community. So this you can think of your genetic information like gene sequence, DNA sequence informations, or PTB, which has structural data for proteins and so on, right? So original biological data, for example, GenBank, your European Molecular Biology Laboratory, that is EMBL, the DNA Data Bank of Japan, that is DDBJ, Protein Data Bank, as I said, and so on, right? And then secondary databases, what they will contain is that they contain processed or manually curated information derived from the primary databases itself. So you're basically going one step further here. And they also include functional annotations, for example, Swiss plot and protein information resource. And then specialized database, they basically cater to particular specific information about particular fields or particular research interests. So for example, HIV sequence database, ribosomal database project. So these are the examples of specialized databases where they have very specialized information, right? And I have also put all of the references here, so you can look through the books or whatever I have cited here as well, right, to get this information. Now, so if we solve a quick question, so as you know, the main one of the primary uh, you know process that we go over in this tutorial classes is that we talk about some concepts that are covered in the lectures for that particular week or some associated concepts, and then we solve some questions, right, which would be similar to the questions that you get in your assignments or in your final exams that will happen later, right? So here what we will do is that in some slides I will have some questions, uh, and then you can answer in the chat basically what the answer to those questions will be, right? And then we'll talk about it. Okay. So here we have a question that is a request for information or data from a database table or a combination of tables. What is that known as? So that's the request for some information from a database, right? So if you remember in our earlier slides, we went over some terminologies, right? For example, ontology, schema, stuff like that. So what term would indicate the answer to this question? So take a little bit and try to answer in the chat, and then we'll look into it. Okay. So this is basically called a query. Okay. So whenever you are trying to retrieve some information from a database, you give a query. As you talked about earlier, it is very important for databases to facilitate easy retrieval of data, right? And that can you can give query. You can give like simple queries, for example, just retrieve some names or some content, for example, some sequence or some organism names. You can search through those, right, in a sequence database, for example, right? However, you can also give a combination of like complex queries, which will help you like really narrow down the search basically through the entire database. So that is called a query. Okay. Now another database related concept which is quite important, uh, that is a language called SQL. Okay, SQL or SQL. So you might have heard this programming language used in the creation of databases. So what does SQL or SQL this term stands for? So try to answer in the chat. Look at it. So I see one question. Can you get this PPT slide for reference? Yes. So I believe from the NPTEL platform of the course, where the course is. So I think there is an option for the spreadsheet where we have uploaded the links to the slides or a Google Drive folder, as well as when we finish uploading the videos for particular lectures on YouTube. So those links also will be there. in that spreadsheet. I think it has been shared on the course platform, course tab on the NPTEL platform. So if you refer to the spreadsheet, you will get the recorded videos in YouTube, and also the slides for every class as it happens. Okay. Yeah. So SQL essentially stands for Structured Query Language, right? Now, what it is, it's basically a specialized programming language that is used for managing and manipulating relational databases. Now, that is something also we'll look into it. Uh, it's a standardized way to interact with the databases, regardless of their underlying DBMS. And then categories of SQL commands. So since it's a programming language, it will have a lot of commands, right? Uh, so they can be categorized into data query language, data definition language, data manipulation language, data control language, and so on. And some of the common commands include, for example, select, which is for retrieval of data from one or more tables, insert, adding new records to a table, update, which is modifying existing records, delete, removing records, create, that is establishing new database objects, alter, drop, and so on. So here I have put the link to an online tutorial, which you basically uh, copy this link or just go to this link. Yeah. So here in this website, you can basically go through like a very basic SQL or SQL tutorial where you will learn about some commands and so on. Okay. This particular website has, as you see on the tabs here, so basic tutorials for a lot of programming languages which you might be interested in. I have given the link for the SQL one. Okay. So that's something you can do. Uh, in the meantime, we'll go to the next concept. Okay, now database management systems, right? Now, what are database management systems? Database management systems are basically software programs which have been developed to streamline every process related to uh, the basics of databases, the foundation of databases. For example, organization, search, retrieval, etc. And they not only stored raw records, but they also have operational instructions to uncover the underlying connections among data. And the primary objective is to facilitate efficient execution of searches, merging different records, generating search reports, and so on. And basically, they can be categorized into two main types. Firstly, relational database management systems and object-oriented database management systems. Now, combining these two, also you can get hybrid, which comprises of both the concepts of relational and object-oriented database management systems. Uh, but when you use a relational database management system, so those databases you can refer to as relational databases. And when you use conversely the object-oriented ones, so those are termed as the object-oriented databases, right? Now, what are these relational database management systems? I think this concept was covered by Professor Bromiha in the first week's lectures. So, can somebody remind me what relational database those are? Can try to answer in the chat. So, what is it meant by a relational database? If you remember the earlier lectures, the relational databases are basically databases where different tables are linked together by common attributes, right? So if we come to that concept, so it's basically just employing multiple tables for data organization. And here, each tables, they are termed also as relations. They have rows and columns. And the columns, they indicate particular fields. And then the rows contain the specific values of that field, right? And these are basically the records in the database, right? uh, and those are indexed based on shared characteristics that are called an attribute. So you can think of it as that different tables, they are connected by at least one common attribute so that you can facilitate search faster from one table to another. So indexing facilitates cross-referencing of data present in other tables, and the system selects linked data from different tables during the execution of the query. Uh, and then the combined informations from all of the tables, they are resulting in an unified report, right? And this basically allows faster access to the specific information. Now, flat file databases are basically when you record the data just in a complete text format where there is no hidden instruction available, right? And these flat file databases, those were used at the very beginning of databases. But for the computer to give you the result of a particular query, it has to parse through the entire database and get the data from here. However, relational databases make that process a lot faster. And how we'll just look at it in the next slide. Okay. And the relational databases, as we saw in the earlier question, is that they use the specific programming language called structured query language, and they have the flexibility where new categories can be added easily without modifying any existing tables, right? So if we look at this slide here, so the very first database that you will notice in the form of the flat file, where uh, they are basically having one, two, three, four different categories, right? These are name, states, course number, and course name. So imagine this data is coming from some university or some course. I think some noise is coming. Okay. Uh, so basically, you can think of it as data from certain ports, right? Where you have the student's name, states, course number, and then course name also. So let's say the data for the courses from a particular department, right, or a particular institute. So you can think of it that way. And then here you will see that each of the entries, they are kind of separated by this uh, vertical line, kind of like a delimiter or a pipe, right? And then here you will see each data records that will go to this four specific attributes, right? So those are separated by these vertical lines or delimiters, right? And this way the entire database is organized. However, you can imagine that if you want to select any particular query, or if you want to get any particular information, so let's say like from the states of Texas, or from the state of Texas, the students are taking which course, right? If you want to keep some complex query like that, or some complex information you want to retrieve, so the computer has to, or the algorithm has to go through the entire file to get the data. However, what the relational databases, they do is that uh, they firstly divide the entire data through in the form of a table, basically, where the different columns, they represent the attributes, and then the rows have the recorded values, right? Now, these different tables, table A, table B, and table C, you will notice that these are linked by some common attributes, like some common columns, right? For example, table A and table B, those are linked by the students, right? The student's ID, you can say. And then table B and table C, those are linked by the course ID, right? So when you give the query to this relational database that, okay, so give me the courses that the states of Texas, the students from the Texas state, they are taking. So what it will do is that firstly it will search in table A where the state information is located. So it will search, okay, so from state of Texas, let's say student 1 and 5, they are taking the courses, right? So they are from the state of Texas. And then it will correspond that data in table B where it will look for what the students 1 and 5 are taking as course. Now, when it gets the course code, then it will search for the course name, right? So math 172 and then biology 689. And then it will get the information like bioinformatics and calculus. Now here you can imagine that the search time was reduced by a lot because in these later tables, the algorithm did not need to go through the entire sort of files. So when it got the information that which students we are looking for, so from this column, students 1 and 5, so it can just correspond that data, the available records for student 1 and 5 through the other tables. So this will reduce the search time by a lot. And in contrary, you can say for the flat file, if the same information was present, so it has to go through the entire file. Now, if you imagine, this is a very simple sort of example of a database. If you imagine, let's say there are hundreds of different columns representing some sort of data, and then there are thousands of records for each of those columns, right? So you can imagine that going through that entire file will take a lot of time. It will take a very high degree of or high amount of computational resource, right? So the relational database, this sort of format sort of cuts down that computational cost, and the time also is reduced by a lot, right? So that is the basic concepts of relational databases. You can get this information from the book Essential Bioinformatics, which I have referred to here. Now, if you look at the object-oriented databases, so these are basically an advancement from the relational databases in one way, you can say. So they basically facilitate representation of complex hierarchical data between different items. Uh, and whether in relational databases, they might not adequately describe the data connections, the object-oriented databases are good at those, right? So basically here what happens is that they store data as objects. You can think of objects as kind of containers that have different data points. And then combining the data and mathematical routines in a unit, the object-oriented databases do. And then the programming languages that the object-oriented databases used are object-oriented programming languages like C++, which links objects using pointers, which basically defines some predetermined relationships between, right? And then the searching involves navigating through objects with the help of pointers. And these sort of databases, they allow more flexibility, allow data to be structured in a hierarchical fashion, and then they also simplify some programming tasks with complex relationships. However, they also sort of lack the rigor of the relational databases or the mathematical rigor in a way you can say. So some hybrid approaches have been taken where the relational form of databases and the object-oriented forms, those are combined to create some hybrid databases, right? So if we quickly go through the example, so this is kind of what an object-oriented database would look like. So here, for example, the state objects and the student objects and the course objects are there. And then what it has to do is that from the state of Texas, it just has to look for the links that it is pointing towards which are the student objects. And conversely, subsequently, those student objects are pointing to which course objects. So it can follow these pointers and then get the information. So those are the object-oriented databases. Okay, so that covers the databases. Now, in the latest lectures, I believe Professor Bhumi covered some basics biochemistry related to proteins and so on, right? So we'll cover some questions regarding those. So here in this slide, I have one question, which is that what are the building blocks of protein? So the building blocks of protein are what? Are termed as what? Are they nucleotides? Are they amines? Are they amino acids? Are they ribose sugars? So take a little bit of time to answer in the chat. So the building blocks of proteins, as we know, those are called amino acids, right? What are nucleotides? Nucleotides constitute what? Which biological macromolecules they constitute? Nucleotides are the fundamental building blocks for nucleic acids, right? Your DNA, RNA, those, right? And the ribose sugars, uh, again, you know that deoxyribose is a part of DNA, and then the ribose sugar are part of is a part of RNA, right? So those are also coming in the nucleic acid formations, right? Okay. So let's solve another one of these questions, which states that pyrimidines have how many aromatic rings? So do they have no aromatic ring, one aromatic ring, two aromatic rings, three aromatic rings? So also try to answer in the chat. Put uh, all of you mention in the chat some examples of pyrimidines and also the examples of purines. So if we look at the structure of pyrimidines and as well as purines, so you notice that uh, purines, they have these two ringed structures, right? And pyrimidines, they have this one ring structure. So here the answer will be one aromatic ring. And for the examples, you can see that adenine and guanine, those bases are purines, and then cytosine, uracil, and thymine, those bases are pyrimidines, right? You know the thymine is found in DNA, and then uracil is found in RNA, right? Okay. Another question is here, which asks which amino acid is predominantly present in the mammalian proteins. So is it L-alanine? Is it D-alanine? Is it L-methionine? Is it D-methionine? Okay, I see hand in the chat. So you could unmute yourself and ask if you have any questions. So if you have a question, you can unmute yourself, and then we'll look at the answer to this question. Okay. So which amino acid is present predominantly in the mammalian proteins? Try to answer in the chat. Let me look at it. So here the answer actually will be L-alanine. Okay. So here the steric relationships of the stereoisomers that are mentioned here. So for example, the comparison is with the L and D glyceraldehyde, right? Uh, so basically, here the carbons of the molecules are numbered with the terminal aldehyde or carboxyl starting from those atoms, and then 1, 2, 3 from top to bottom. That is what is represented here. And the R group of the amino acid, for example, in the case of alanine, that would be the CH3 group. That is the R group here, right? That is always below the alpha carbon. And the L amino acids, they basically represent the amino acids where the alpha amino group are on the left. Okay. And for the D amino acids, they represent the amino acids which have the alpha amino groups on the right. So the particular stereoisomer of alanine that is predominant, that is the L-alanine. Okay. So if we move to the next question, uh, this question again asks about the side chain or the R groups, right, for the amino acids. So which amino acids they contain aromatic R groups? Are those isoleucine and tyrosine, tryptophan and threonine, leucine and phenylalanine, or tyrosine and tryptophan? So aromatic R groups. That means that the side chain that will have a ring structure, right? An aromatic ring. Try to answer this one in the chat. We'll look at it. So the answer to this question will be tyrosine and tryptophan, right? Now, if you look at the categorization of the different amino acids, so basically you can broadly divide them into non-polar, aromatic, polar, positive charge, or negative charge R groups, right? R group containing amino acids. So here, if you look at this particular box here, so you'll see that phenylalanine, tyrosine, and tryptophan they contain aromatic R groups, right? And then conversely, you might be asked some questions like which amino acids have positively charged R groups. So those would be lysine, arginine, histidine. Negatively charged R groups would be aspartate, glutamate. And then the polar ones will be like serine, threonine, cysteine, aspartame, glutamine. And then these will be the non-polar aliphatic R groups, right? So these sort of uh, categorization of different amino acids or their polarity or hydrophobicity, you might get some questions related to this sort of concepts. Okay. Now, a very similar one is that you might be asked to rank certain amino acids with increasing order of polarity or decreasing order of polarity. So some sort of ranked order, right? So here the question is basically the same one. So between all of these options, so which one is ranking the amino acids with increasing polarity? Okay. So again, try to answer this one in the chat. So these will be the three structures of the amino acids. And we notice, of course, the only difference are the R groups, like the side chains. So here the aspartate, you can see it has a CH2 C double minus, and then for the serine you have a CH2OH, and then for the threonine you have a CHOH and then CH3. Right. So based on that, what do you think the order of polarity will be for these amino acids? So here the polarity order for the functional groups, it goes something like that. Okay, so something like this. So firstly you have the amide groups, then the carboxylic acid, then alcohol, then ketone or aldehyde are roughly similar, then amine, ester, ether, and then finally alkane. So based on that, okay, here you see one CH2CO minus for the aspartate, right? So for the increasing polarity, if we order, the aspartate would come at the very end because it should be the highest polarity among all the options that are available here. Now for the serine and threonine, you will notice that both of them have an OH group, right? However, for threonine, you will notice that it has a slightly longer side chain than the serine because there are two carbons here, right? In the side chain, you will see there is CHOH and then CH3 also, whereas in the serine, it was only CH2OH, right? So you'll notice that the alkanes, they come at the very last, and the number of carbons in the side chain or number of carbon chains that are present in the side chain R group, that also influences the polarity. So the more number for threonine essentially leads to a decreased polarity for threonine if you compare against serine, for example, here, right? So here threonine will come at the very first, then serine, and then aspartic acid if we rank it in an increasing order of polarity like that. Okay, so this polarity order of functional group, this might be something to remember. Okay, and here if you look at this chart, so again we see the similar sort of classifications for the amino acids, like non-polar, aromatic, polar, positively charged and negatively charged. Uh, so alongside the various parameters such as the pK values, the abbreviation, the occurrences in proteins. So for example, here you will notice the percentage occurrences for all of the proteins. So here you have something called the hydropathy index. So can anybody tell what this hydropathy index indicates? You can answer in the chat. So the hydropathy index is basically another parameter that can help us categorizing the polarity of the side chains for the amino acids, right? So here, what it is that these values reflect the free energy of transfer. So the del G of transfer uh, of the amino acid side chains from a hydrophobic solvent to water. Okay. Uh, so could you answer a small question that if a side chain of an amino acid is polar, so whether the transfer from a hydrophobic solvent to water will be favorable or not?
Yes, sir.
Yeah, it will be favorable, right? So that's why, since the del G, the transfer will be favorable, so for the polar groups, the del G will be less than zero. So negative value basically indicates that some sort of amount of energy is liberated, and that will basically mean that the end state is of a lower energy, right? And lower energy will mean higher stability. So that sort of one concept is being applied here, right? So for the charged or polar amino acids, it will be favorable, and then for the others, it will be unfavorable, right? For the hydrophobic ones. So those are indicated by the positive values, right? So you can look at this hydropathy index, and wherever you see negative value, you can see that those will have the favorable transfer from a hydrophobic solvent to water. So you can look into it, you can look at it, and then you can also categorize the order of the polarity in that way also. For example, in positively charged or negatively charged R groups, you can see a very high negative value, right? And for the polar uncharged ones also, you can see like very high value here. And even though cysteine, that is having a positive hydropathy index here, the point is that it is also considered generally as a polar amino acid, and that is because the sulfhydryl or the SH group that can act as a weak acid and can form weak hydrogen bonds with oxygen or nitrogen. So that is why, despite having a positive plus 2.5 value of the hydropathy index, it is also categorized under the polar groups, polar R groups. Okay. So, which of the following residues can form hydrogen bond with the side chain atoms? So, try to answer in the chat. So here the answer will be serine. Okay, serine forms hydrogen bonds with the side chain atoms. Because if you look at the structure of the serine amino acid, you will see that there is an OH here present at the side chain alpha, right? So that will allow it to form hydrogen bonds. Yeah, so it will be serine. Now, I believe Professor Gumiha went over some of the concepts regarding the functions of proteins, right? Uh, so the structural proteins and the functional proteins also he covered those concepts. So here the question asks, for a channel protein, what will be its main properties? For a protein to function properly as a channel protein, what characteristics does it require? So firstly is gating, then there is selective ion conduction, there is both, or conversely none of the above. So you can try to answer in the chat. Then we'll look at it. Okay. Here the answer will be both. So what gating is basically that these channel proteins need to be able to change its conformation so that channel will be open or closed in certain situations, right? So basically they can gate the flow of ions from one side to the another. So that is gating. And then selective ion conduction is that they need to allow certain ions only to pass through, right? So basically selection of ions. So that is selective ion conduction. And for the channel protein to be functional, it needs to be both. Okay. Now another question here, which asks which of the following is not true about the secondary structures of proteins. So here the first option is the hydrophilic or hydrophobic character is important to the secondary structure. The ability of the amino acids to form intramolecular hydrogen bonds, that is important. Alpha helix, beta pleated sheet, and beta turns are examples of protein secondary structures, or all of the above. So remember, it asks which one is not true for the protein secondary structures. So try to answer in the chat. Look at it. So I see a lot of A's and some D answer in the chat. So here the answer basically will be A. Okay. Uh, so if you go through the other ones, intramolecular hydrogen bonds, that is very much important to the secondary structure of proteins, because if you remember alpha helix beta pleated sheets, those have particular pattern of hydrogen bond formations, right? And of course in C you see in the option C you will see that the alpha helix repeated sheets and beta turns, they are obviously the protein secondary structure examples, right? However, the hydrophilic and hydrophobic character of amino acids, that is much more important in forming the tertiary structures of proteins, the 3D structures of proteins, right? And the higher order up and onwards from that, right? So that is basically where the hydrophilic and hydrophobic character comes as the most important part. Now, this is basically an example of the amino acid chain. And here you will see some dihedral angles or torsion angles, you can say, those are indicated. So for example, the NC alpha, you will see the rotation around this bond is indicated as phi symbol, and then the C alpha C, that is the psi symbol, right? So here one plot is represented where one axis you see SI in terms of degrees, right? And also in the plot you see some areas are indicated or highlighted here. So does anybody remember what this plot is called? This sort of plot. So the x-axis is kind of not mentioned here. I think it's cropped probably, but it should be the phi in degrees. Okay. So phi versus psi. What plot is this? Yeah. So as I see, I think in the chat, it is called the Ramachandran plot, right? So basically, which of these phi versus psi data points that are energetically favorable, right? For example, in different secondary structures, you will see that the favorable regions lie at certain different phi and psi value ranges, right? For the parallel beta sheets, you see it here, for the anti-parallel here, right? For the right-handed alpha helix here, and so on. So why this sort of preference in the phi and psi angle occur? It's basically because the secondary structures have certain patterns. So these phi and psi ranges basically prevent the steric hindrance, you can say, from happening, right? And so they have a minimized sort of energy state in a way. So that's why a certain type of secondary structure patterns, they favor different phi and psi ranges. Okay. And again, here the same is represented. So idealized phi and psi values. Obviously, it is not the exact point as the idealized values. That does not happen always. There will be a range, obviously, but you can kind of refer to this table as what should be the ideal value for phi and psi for different sort of secondary structures. Okay. Now here we have one question. So which one of these can best describe the reason for the association or clustering of hydrophobic residues in aqueous solution? So in the aqueous solution, if you just put a sort of stretch of amino acids, so they will form a cluster together, right? They will come together and then they will form some higher order structures, right? The tertiary structures. So which of the following forces basically contribute to it the most or contribute to it essentially, right? So is it the formation of covalent bonds, electrostatics, entropy related increase, or hydrogen bonding between amino acid residues? So what would be the most prevalent force that dictates the hydrophobic clustering? Try to answer in the chat. Here, essentially, in the hydrophobic clustering, the concept of entropy comes. Okay, so the answer for this question will be a favorable increase in entropy. Now, can somebody mention what does entropy indicate? You can mention in the chat. So I see one answer, that is randomness. Yeah, it's basically the state of disorderedness or the measure of disorder in the system, right? Now, what happens is that if I can here, so let's say you have in a solvated system or in a solution, right? So let's say you have put the amino acids here, right? Now, a very important property of water is their efficiency in forming hydrogen bonds, right? So the waters are linked through those, and that is basically a very highly ordered structure, you can say, in a way. Now, when you put the amino acid sequences, you can say, or the amino acid stage of amino acids in the water, what will happen is that around each amino acid, the water will try to go back to that ordered nature that it was before. So it will basically form a shell of water, which is also called a solvation layer in this case. Okay, so it will basically form a solvation layer around the amino acids, and this solvation layer, so the waters that are part of the solvation layer, they are very highly ordered, essentially. Okay. Now, if you compare it to another state where, let's say, the amino acids are not far apart, but the amino acids are overall, let's say, forming some sort of clustering or clump here. Now, here still you will get a solvation layer, a layer of water or shell of water around this sort of clump or cluster. But if you compare the volume or the surface that is covered by the water in the case of where the amino acids are far apart, and then the case of where the amino acids are clumped or clustered together, you will basically notice that during the clumping or clustering, a lot of parts of the amino acids, they are kind of get buried, right? They are getting buried. So what will happen is basically that the combined volume of all of the solvation layer when the amino acids are far apart, the combined volume of the solvation layer will be higher in this case, right? And then when the clumping occurs, that combined or that solvation layer volume, it will be slightly lower because a lot of portions of the amino acids, they are getting buried inside the cluster, essentially, right? Now, as we know that from the second law of thermodynamics, you know that for spontaneous processes, the entropy of the universe increases, right? So increase in entropy is favored, right? Now, since as we talked about that the solvation layer is kind of ordered structure, so basically the decrease in the volume of solvation layer will be favored because that will be kind of less ordered in a way, right? So that is why the state from this to this is favored because it is an increase in entropy. And the increase in entropy comes from basically the fact that the entire volume of the solvation layer or the surface of the solvation layer that will be reduced here. So since the solvation layer represents the ordered structure of waters, so reducing that the volume of the surface of the solvation layer, that will uh, essentially cause the disorderness to increase. So that will be a favorable increase in entropy. So that is sort of one of the primary force that is contributing to the clustering of hydrophobic residues in aqueous solution. And stabilizing the structure. So that is where the electrostatic interactions, like the non-covalent interactions and then hydrogen bonding, etc., that comes into play. But the primary force here will be related to entropy. Okay. So this is kind of another knowledge-based question. So which of the mutations they lead to sickle cell anemia? So try to answer in the chat. Then we'll look at the answer. Yes, I see some answer B's in the chat. And yeah, basically the glutamic acid seeks to valid in hemoglobin that leads to the cause of sickle cell anemia. So again, these sort of knowledge-based questions you might encounter in the assignments as well as the exam. Now here again, something related to the side chain R group. So which amino acid has the side chain imino? So you can try to answer in the chat. Because here the answer or the structures for the amino acids are kind of like this. Uh, and basically, this portion that you see in proline, right? The NH2 plus here. So that is called the imino group. And here, the correct answer for the question will be proline. Okay. Now, another one question that is here is that which amino acid cannot rotate 10 polarized right? So can you try to answer this one in the chat?
Sir,
Yeah.
all amino acids are optically inactive except glycine. But glycine is not given in the option.
Yeah. That was kind of the question here because glycine should be the one, right? Because it has you see 2H attached to the carbon, and so that will make it optically inactive, basically, right? So glycine was, yeah, not given as the answer intentionally. So for this one, glycine will be the proper answer. That's correct. So here another question we have is that which of these following are structural proteins? So I'll just quickly go through some of these questions, I guess. Uh, so as Professor Gomiha might have mentioned, that globular proteins are basically functional proteins, and then structural proteins are your fibrous proteins, right? So here in this case, the answer will be fibrous proteins. And have some concepts regarding alignments to cover as well. Uh, so we'll be starting starting on the concepts about alignments. So when Professor Gromiha introduced alignment in this section, sequence alignments, you might have heard about gaps in the alignment, which are also associated with some sort of penalty scoring, right? Gap penalties. So what does a gap in an alignment represent? So are they due to insertions, deletions? It can be both insertion, deletions, or none of the above. So in sequence alignments, these gaps, they essentially represent one term you might have heard of, that is called indel. So indel means both insertions and deletions. Because the gap penalties as well as the scoring related to those, they do not essentially in the sequence alignment, they do not see whether one sequence came before another or not. So which one came first, which one came second, those sort of concept regarding that is not applied here. So both insertions and deletions, they are kind of classified under just one category, which is called indel. So that is what a gap represents. Okay. Now, so we'll start on the concepts of alignments. So why do we need to align sequences? So basically, what alignment tells us is that the comparison between different sequences, right? In which sections they are similar, or which two sequences are more similar than other two pairs, right? Those sort of things. So why do we need to compare biological sequences in this particular way? Why is there a need for sequence alignments? What sort of functions do they serve? So can somebody mention any points regarding that? So one, I think in the chat, yeah, conserved domains. That's correct. So basically, if you look at the broader applications of sequence alignments, so you can obtain meaningful insights into functions or mechanisms for a sequence from alignment, where you are aligning with another sequence which is potentially better characterized, right? So if you find, for example, in your lab you are encountering a sort of new nucleic acid sequence, right? Some new sequences is coming up and you want to see, okay, what sort of function does this have? Then through alignment, you find another sequence which is better characterized, and you can infer, okay, so this better characterized sequence, this has this sort of function. So you can infer that this newly found sequence that will probably have similar functions, right? So characterizing newly found sequences from a potentially better annotated ones, right? And then again, if two, let's say proteins, they are evolutionarily related or not. So those sort of insights can be found from sequence alignments, right? Evolutionary distance can be found or can be inferred from multiple sequence alignments and so on, right? And then structurally or functionally similar regions in proteins, you can say. So that is also the concept of motifs, right? So different conserved motifs across a class of proteins you might be able to identify through sequence alignments. So these sort of implications are there for sequence alignments, right? So protein structure prediction, annotations, then let's say assembly of sequence into larger construct of genomic sequences, mapping, etc. Those sort of functions, those sort of processes require sequence alignment in the broad scale. Uh, Now, one very basic method of sequence alignment, which Professor Brumia covered, that is called the dot plot, right? So the dot plot is basically if you, let's say, put two sequences on the x and y axis, and you put a dot on the matching characters, right? Wherever there you will find like the x and y axis characters match, so you put a dot in there. And then when you finish constructing the dot plot, you can infer some information from the let's say diagonals or wherever you are finding matching set of subsequences and so on. So here the question asks, so when constructing dot plots using these two given sequences, where these continuous dots are observed. So let's say in this sort of grid, you are putting these two sequences, right? On the x-axis, you have LEADER, and then on the y-axis, you have ADERFF. Okay. So more than three continuous dots, they are being observed at which area uh, of the matrix, you can say. below the diagonal or above the diagonal, or are there no continuous dots at all? So here, to get the dot plot, basically what you have to do is that you have to find the matching set of characters, right? So for example, uh, here the first dot, let's say you can put at a place like this, where E and E are matching. Then another one is here, because again you see another E and E. Then here you can put like A and A for both of these sequences, they are matching, right? And then here you will see the aspartate and aspartate matching. And then here you will see two glutamates and glutamates matching together, right? So you will see this diagonal, that is a continuous set of three dots in the uh, form of a basically line, you can say. So that is forming below the diagonal. So diagonal is basically, in this case, it will be this. Okay. Now, if say we had the same sequence on the x and y axis, you will notice that the matching set of dots will appear directly at the diagonal. However, here that is not the case, and instead, okay, so another matching was there, so R versus R. But here, in this case, you will see that these continuous dots, they are appearing below the diagonal. Okay. Now, what does this sort of dot plot indicate? So, in detail, uh, the basic concept is that sequences A and B, they can be represented on this matrix. Earlier we put like x at the bottom and then y at the left, but here the method is in an alternative way, you can say. So x at the top and then y at the left hand side, right? So put the two sequences in this fashion, and then you just fill the matrix with dot where the identical characters are present, right? So starting from the first character in B, each character you can compare through the entirety of A sequence, and then a dot is placed everywhere to find the match, and then the process is repeated for every subsequent characters in B. Uh, and then whenever you see that there is a diagonal rows of dots on the matrix, as we saw in the earlier example, so those will be the matching sort of subsequence in the entire sequence, right? And then isolated dots, they most likely contribute to random matches, or you can say noise in the signal, and they do not indicate any significant alignment properties here. And the point of the dot matrix is it's a very basic visual representation of the sequence similarities and differences. And here you have one example of dot matrix for the amino acid sequences. Okay, of this fudge lambda and then fudge p22. Now, so the process, as we mentioned earlier, is that you put one sequence for the x-axis and then another one at the y-axis. So here you put the first one at the top and then the second one at the left, right? And you just put dots in there, right? So you put dot at the intersection of A and A here, then dot again here because A and A are matching. Then C and C are matching in two places also. Then G is matching at only one place here, and then so on. So here you get the dot matrix, right? And throughout the construction of the dot matrix, two parameters, they come into the picture, which are the window sizes and stringency. Now, if we just go back to the earlier picture here, you will notice that if you sort of match or put a dot on every matching character, then the whole plot will be a lot noisy. So to infer any sort of significance about the similarities from the dot plot, you will need to identify this particular stretch of diagonals, or uh, where basically the consecutive dots are present. But if you start putting everywhere when the character is matching, it will get a lot of noise also in the plot. So to sort of relieve that condition, one concept is can be introduced throughout this dot plot, which are window sizes and stringency. Now, window sizes are basically, instead of comparing each character, like one character to another character, you put a sliding window, right? So you put a window, let's say window size of 3, you compare 3 characters at once. So for example, so here, obviously, what we did, it had a window size of 1 and stringency of 1. So we are comparing each character, and stringency 1 is basically means it is very strict for comparing the each characters. In this particular case, it doesn't matter because the window size is 1 only. But let's say we had a window size of 3 instead, right? So if we had a window size of 3, what we would be doing is that for the x and the y axis, we'll start comparing 3 characters at once. Okay. Now here in this case, if we compare this ACG versus ACA, of course, the entire sequence is not matching, right? So that is where the stringency comes. So the stringency is basically the strictness of the comparison. So if you say have a stringency of 2, then what will happen is that if 2 of these characters in this 3 element window, if 2 of those are present at the exact same position, then you can say that they are matching and you put a dot here. So for example, if you compare this first two 3 element windows on the x and the y axis, you will see that A and C, they are coming at the very first in both of these cases, right? So that means if you have a stringency of 2, you are able to put a dot here. Okay, in the case of stringency of 2. Now, if you had, let's say, stringency of 3, so that means it's very strict. It has to match all 3 of the characters for both of these windows. Then in that case, you would not be able to put the dot here because obviously A, C are matching, but the third character A versus G is not matching, right? So that is the concept of window size and stringency. And when you are done with, let's say, comparing ACA versus ACG, uh, then you move to compare, let's say, ACA to the next overlapping window with overlapping characters. So the next window, for example, you will compare is this one, the CGC versus ACA, and then you will decide whether to put a dot in the intersection or not. Okay, so that way you have to compare the entire thing. So if we just go through the example of window size of 3 and stringency of 2, again, you compare ACA versus ACG at the top, and you find that the first 2 characters are matching. They are present at the exact same location. So then you put a dot here. The next thing you will do is that you will continue the match throughout like this. So you will compare like CGC, and in the next step you will compare GCG with ACA, right? And if you find the match, you will put a dot here. And after doing that, you will then move to the next set of 3 characters here. And you will consider that as a single window. And then again, you will compare the window on the other axis and overlapping windows on the other axis based on your size of the window. So basically what we will do is that we will find any other matches that are present there. And if they are matching, we put a dot here. So for example, here in CGC and CAC, you see the first and third elements are matching. In this case, this one and this one matched. So you put a dot at the intersection, and then you find any other matches in any other windows or not. So here you see also another match. So this ACG versus ACG, obviously it is matching entirely. So you basically put a dot at the intersection, and then you compare all the windows and finally find that you find another match here, right? ACG versus GCG. So here the second and third elements are matching CG. Okay, and then when you are done constructing the dot plot here, you will notice that it has a much fewer dots than if you did it with window size 1 and stringency 1. So it has fewer dots, right? So basically you can imagine that if you're comparing large sequences, two large sequences, let's say thousands of amino acids long, right? 1,000 amino acid long, let's say. So if you did that with window size of 1 and stringency of 1, what you would have done is that you will have put a lot of noise in there because you have to put the characters everywhere, right? You have to put the dots at empty matching places. But if you increase the window size and then sort of figure out a proper stringency, then you can you can basically reduce the noise of the plot, and then you can maybe possibly infer some better information from the dot plot.
Sir?
Yeah, please.
Sir, how stringency is 2 here?
So in this case, so the window size 3 is mentioned by this rectangle, right? Now stringency 2 means that out of these 3 elements of the window, when you are comparing two windows, two at least two characters they have to be matched at the exact same locations. Okay, so for example, in this one, if you compare GCG versus ACG, you will see that while A versus G, that is not matching, CG and CG are present at the second and third place, right? So that means two elements are present at the exact same location, you can say, or exact same places. If you have like three different slots, first, second, and third slots. So on this window also you see C and G are present at the second and third slot. On this window you also see C and G are present at the second and third slot. So you can basically put a dot here. So stringency is basically the strictness of the match. So if you put a stringency of 3, then you have to be matching all 3 characters exactly, and only then you can put the dot. If you have a stringency of 1, then one character matching is fine. You can put a dot there. So you have to basically play around with the stringency and the window size in some actual sequence alignment processes, visualizations. Okay. So stringency 2 means exact two locations in this 3 element window, they have to be matching exactly. Okay. Uh, so here, basically, again, is an example of dot matrices that can be formed from amino acid sequences and also from nucleic acid DNA sequences, for example, on the right side. Okay, and here again, as some additional resource, I have put a link here. So this link will basically lead you to a website. So this particular website here. So this is made by Barry Grant from the Grant Lab. So they also make some visualization tools for R and so on. So one of their visualization tools is called Bio3D, which is very applicable in some sort of analysis. Okay. So here basically what it has is that first you have a protein dot plot and then you have a DNA dot plot. And just for a visualization exercise, you can increase, for example, the window size. Then here you have some additional parameter that is called window step size, right? And then you can just play around with that. And let's say at some point, you can just try to see where, at which of these window size, the stringency parameters. So you can try to see which of the combination of these parameters can give you better visualization, reduced noise, and so on, right? So you can just play around with that. The link will be available in the slides. Okay. Now another question that sort of can arise here. So let's say if you have a 1,000 element long, 1,000 nucleotide long DNA sequence, you are matching against another 1,000 nucleotide long DNA sequence, right? And conversely, let's say you have a 1,000 nucleotide long, 1,000 amino acid long protein sequence, and then you are matching against a 1,000 amino acid long protein sequence. So basically DNA versus DNA. and then another one is protein versus protein dot plot. So in which of these cases do you think the noise would be more? So a DNA sequence versus DNA sequence matching, and then a protein sequence versus protein sequence matching. So if you construct a dot plot, in which of these cases do you think or do you expect the noise to be more? I see one answer, that is DNA. So if you basically think about it, you will see that the DNA only has 4 nucleotides, right? ATGC. Whereas proteins, they have 20 naturally occurring amino acids, right? So basically, for any particular set of nucleotides to be matched, since the nucleotide number, the overall nucleotide number is very low uh, as compared to the total number of amino acids, so you'll see a lot more noise here because you will find more frequent matches in the nucleotide versus nucleotide comparison or dot plot formation, right? Because you will only have 4 nucleotides on each axis, so you'll encounter more frequent matching of characters there uh, as compared to the proteins where you have 20 different amino acids. So the matching basically will be sparse, you can say. Okay. Uh, Now here is one other dot plot. Now this particular one is basically, let's see, this particular one is basically comparing the same sequence against the same one. So on the left hand side you also have the same sequence. On the upper side also you have the same sequence. And when you sort of manipulate the window size and stringency, you can see some sort of pattern like this. Okay. Now, what do you think is the importance of comparing the same sequence in a dot plot in a sort of visualization sense? And in the B plot, what do you think these sort of particular patterns where you see some stretch of basically matching characters they indicate? Remember that you are comparing the same sequence uh, on the X and Y axis. And try to answer in the chat. If you compare the same sequence on both of these axes, as we discussed earlier, whenever you find uh, this sort of area of matching sequences, you will notice a pattern like this kind of diagonals, right? And because here you see the same sequence on the X and Y axis, you will see just from the very beginning to the very end, there is a big diagonal here, right? That is you are getting. That is something that you are getting here. Now, alongside that, you see uh, again some match can be found throughout this particular region that I'm highlighting here. Again, So these matching that you are find repeatedly throughout this particular region for let's say this particular subsequence, that will indicate repeats, right? Short stretches of repeats that are present throughout this sequence. Because if you imagine on the x-axis some sequence is present here, some subsequence. So because you are getting match at all of these positions, that means at this particular region also the same subsequence is present. At this region also the same subsequence is present, and so on. So that means that this sort of repeated subsequence is present throughout this part, and that can lend you some idea about repeats that are present in a protein, let's say. So this particular case was the case for a human LDL receptor, and then initially you had this very cluttered plot, right? But after doing it with window size 23 and stringency of 7, these longer repeats they were found by the sliding window method, basically, right? So again, that means that modifying this window size and stringency and trying to identify what sort of leads to a better visualization is also important. And not only comparing different sequences, but same sequences comparison, that can also give some insights. Okay. So another question that we have here is the example for a globular protein. So again, that is sort of leading to the structure and function of proteins, which Professor Krum has covered. So if you remember, two sort of types of proteins were discussed, like fibrous proteins and globular proteins, right? And for the fibrous proteins, those are generally structural proteins, right? So if you remember that the examples included keratin, fibrin, collagen, etc. And these other sort of functional proteins, they are kind of globular proteins here. And the example from the globular protein, that is insulin, that was included in this option. So the answer should be insulin. Now, alongside the dot sequence, just after that, for the alignment perspective, so a sort of scoring system was introduced, right? So let's say in an alignment, if you find the alignment, you give a match score. If you find that the characters are not matching, so you give a mismatch score. And then based on the presence of gaps, you give some sort of gap penalty, right? Now, why is there this complex sort of scoring is needed? Because you need to quantitatively analyze the extent of the alignment, right? And you need to find the best alignment for two sequences. So for some simple sequence, you can be thinking of two like very simple sequences. They can be aligning. They can be found to have been aligned in like multiple different ways. So maybe just the initial part of one sequence is matching with the final part of another sequence. Maybe there is some alignment throughout and some mismatch and match are present. And then maybe you also need to introduce indels in one sequence so that there will be a gap. But ultimately, considering the entire score, the alignment would be better, right? So for that, this sort of quantitative process, a quantitative perspective, so the match scores, mismatch scores, and the gaps or gap penalty associated with it are introduced. Now here for this question, you have been asked that you have already been given an alignment, and based on the match, mismatch, and gap scores given, so what will be the net score of the alignment? So you can try to calculate it in the calculate and report in the chat. So remember, here you have to just count how many matches you get, then how many mismatches you get, and then whether or not there are gaps present in the alignment, right? And for that, you give a gap penalty. Now, why is that? Because generally, throughout the evolutionary processes, the presence of gas is much less frequent. So when you are trying to align two sequences, if you find a gap, so that means that is not that much favored in the evolutionary processes. And that is why for quantitating the score, you give a penalty of a negative value in there. Now, of course, if two characters are matching, then you get the match score. And if they're not matching, so that means some sort of mutation or substitution has occurred there. So for that, you give another negative score, which is a mismatch score. So the process of working through this sort of problem should be, so I see the answer is -1. So you just find out how many matches. For example, here in two places highlighted in blue, we are seeing the matches. So 2 times 2 is 4 for the two matches. Then you have three mismatches. So 3 times -1 equals -3. And then one gap penalty as well, -2. So the net score will be -1. So like very standard numeric problem here. Okay. Uh, the like of which you might encounter in the assignments as well. Okay. Now let us walk through another similar question. So here for two sequences, you have been given three sets of matches. And again, you have been provided the match score of 1, mismatch score of 0. But instead of only one gap penalty, here two gap penalties are introduced. And those are origination penalties and length penalties. And you will notice that the origination penalty is of slightly higher negative value than the length penalty. So again, that is because so the origination penalty is basically if you have a new gap introduced at a particular point, so that will provide a score towards an origination penalty. Because generally what happens is that again throughout evolution, if a gap is probably, let's say at some point a gap is introduced at some part, so another gap, if that is present just after the previous one, so that is kind of sort of you can say less unfavorable than if another gap was introduced after a stretch of match or mismatches. Basically like that. So for example, in the first one, you are seeing one gap is present here, and then after that you are finding some sort of alignment here, right? AT here, and then just after that another gap is introduced. So this introduction of multiple gaps which are not subsequent at different places, that will be less favorable. So that is why you are given basically 2 times the origination penalty at this particular alignment. Similarly, 2 times origination penalty will be given here. And then here, because these two gaps are present just one after another, so only one origination penalty will be given here. Because if you compare the other ones for the, from the gap perspective, so this will be less unfavorable because it is present just at the subsequent location. And then how many gaps are present? So for each of those gaps, you have to give one length penalty score. And for simplicity, again, here the mismatch score is considered 0. So try to work it out, and then you'll see the answer. So I see one question in the chat. So yeah, origination penalty and length penalty are basically relating to the gaps again. So length penalty is the simplest one. It's basically how many gaps are there. Okay. So for each gap, if you give a length penalty score of a negative value. Now, why are you giving a negative value? Because again, throughout the sequence alignment process is basically lending you an insight into the evolutionary processes, right? And the evolutionary relationship between different sequences. So what happens is that uh, throughout the evolution, introduction of gaps in sequences are not favored. Okay. Uh, Again, if you compare, like for example, if there is a mismatch, so that is called a substitution, right? Substitution or mutation. So again, that is also not favored, but the introduction of gap is even more unfavored, you can say, right? So every time you see a gap, you have to give a length penalty. Now, another concept is that the origination penalty, which is basically every time you see the origin of a gap, right? Because again, if you sort of think about the hierarchy, so origin of a gap is even more unfavorable. Okay. So in these first two cases, you see there are two origin points of two gaps, right? So one is here and another one is here. So that is even very highly unfavorable. So for that, you have to give two times the origination penalty. For the second one, again, is the same. For the third one, though, only one origination point was there for the gaps, and then the next gap was just present just next to the previous gap. So here only one time the origination penalty you have to give. So the origination penalty for the origination point of a new gap. And because the origination of a new gap is highly unfavorable, so that's why you give the penalty for every origination essentially, or every origin point. And for the length is that how many gaps you see. So for that you have to keep the total length penalty. So if we just walk through it, for example, in the first one, you will see three matches, right, of alanines. So that will be a total of 3 points. Then you will see three mismatches. So since mismatch is 0, so the total will be 0. And then there will be two origination penalties because at two different points, a gap was originated. So 2 times -2 is -4. And then the net score will be -3. Now for the second alignment, there are two length penalties as well. So the net score will be -3. So for the second one, you see 5 total alignments, right? Sorry, 5 total matches. So 5 times 1, that will give +5, and then one mismatch, that will give 0. And then for two different introduction of new gaps, you will give two origination penalties, so that will be total of -4, and then two length penalties because there are a total of two gaps present. So two length penalties will be -2. So in this particular case, you will find the net score to be -1. And for the third one, if you work it out, so you'll see 5 matches, one mismatches, only one origination penalty in this case because only one origin point of a new stretch of gaps was present, right? So only one origin point, so one origination penalty, and then two length penalties, which will net you a score of +1, right? And the best alignment that will be. So in this case, the third one should be the better one. Okay. So from this example only, you can see that there are different ways that you can align. It is very important to quantify which would be the best, right? So that's why you have to work out via introduction of different scopes. And again, in some later classes, you'll see that every mismatch, for example, so those are not uh, equivalent essentially. So if some hydrophobic residue is being mutated into hydrophobic residue, so for that the mismatch score will be lower. But let's say if an hydrophobic residue is being mutated into a charged residue, if that sort of mismatch is occurring, then you have to give it a higher penalty because again throughout evolution one hydrophobic residue might be mutated to another one, for example valine to alanine, let's say. So that will be again less unfavorable than if the valine was to be mutated into some charged residue like glutamic acid, right? So that sort of way you have to quantify the points. Okay, I think today we'll wrap up the class, and then some remaining concepts we will discuss in the next session. Okay. So again, for the next classes also, Professor Komiha will introduce the more complex sort of concepts like the PAM matrices, Blossom matrices, and then various concepts regarding sequence alignments and so on. So again, we will cover those in the next tutorial classes. And as always, there will be again 2 other tutorial classes that will be done by 2 other PhD scholars on Saturday and Sunday. Particular timings and the links should have been given to all of you. So if you have time, you can join those classes as well. So based on your preferable time, basically you can join the TA session. For each TA session, the recording will be available on YouTube. So we'll upload it on YouTube and we'll put it on the shared spreadsheet. And also the class slides will be available in the Google Drive folder. Okay. So isn't length penalty is for gap which is present after the existing gap. So see, you can so if some sort of scoring exists which counts the length penalty as existing after an existing gap, so that can also be done. So different sort of scorings are there, but for this particular case, and also what Professor Bromiha introduced. So in these sort of scoring situations, the length penalty are considered as every stretch of gap or every single gap that is present as an individual one. Okay, so every individual gap, those are counted towards one length penalty, and every new origination points, those are counted towards every origination penalty. So if you have, let's say, a total of 3 gaps, then you have to give 3 total length penalties. And then let's say those 3 gaps are present subsequent to one another, then you have to only give 1 origination penalty because only one single origination point was present. So in that sort of, but obviously scoring is very complex. So in the next few classes, you will also see that sort of different processes for creating these scoring points. Those have been devised also. So those will come in the form of, let's say, different matrices like PAM matrices and so on. So there we will also see how the scoring occurs between different set of substitutions, mutations, and everything. Okay. Okay. So again, thank you everyone for watching, for attending the session, and yeah, we'll see in the next class. Okay. So the remaining concepts, whatever I have, I'll cover in the next class, and then for the next week's lectures also I'll cover. Okay. Thank you everyone.
Thank you, sir.
Yeah. Thanks.